Welcome to G Fun Facts Online. Imagine holding a piece of a 40,000-year-old woolly mammoth frozen in the Siberian permafrost. For decades, we could learn about its life from its bones, and later, from the genetic blueprint stored in its ancient DNA. But what if we could go deeper? What if we could read the very script of its life, eavesdropping on the bustling activity inside its cells in its final moments? That's not science fiction. It's the electrifying new field of paleotranscriptomics, and it's changing everything we thought we knew about resurrecting the past. Today, we're decoding the fragile messengers that bring ancient life into vivid, dynamic focus. For a long time, studying ancient life at the molecular level meant one thing: paleogenomics, the study of ancient DNA. This field has been revolutionary. It's how we know we share DNA with Neanderthals, how we've mapped the genomes of extinct giants and traced the spread of ancient plagues. The 2022 Nobel Prize even went to Svante Pääbo for his pioneering work in this area. Think of DNA as the master cookbook in a kitchen. It contains every single recipe the kitchen could possibly make. It's the organism's ultimate potential, stored in a tough, stable double helix structure that can survive for hundreds of thousands of years. It tells us what an organism could have been. But paleotranscriptomics does something different. It doesn't look at the cookbook. It looks at the recipe cards being used by the chefs at the exact moment the kitchen closed. It studies RNA. RNA molecules are the active messengers transcribed from DNA. Messenger RNA tells the cell which proteins to build, while other RNAs regulate which genes are turned on or off. The collection of all these active RNA molecules is called the transcriptome. By sequencing the transcriptome, we get a snapshot of what the organism was actually doing. Was it cold? Was it fighting an infection? Was it starving? We're no longer just looking at the genome, we're looking at what the genome does. This sounds incredible, but for decades it was considered impossible. The very thing that makes RNA so useful, its active, transient nature, also makes it ridiculously fragile. There are three main reasons for this. First, the sugar in RNA's backbone has a chemical component that makes it highly susceptible to breaking down in water. Second, RNA is usually single-stranded, leaving its genetic code exposed to chemical and enzymatic attack. DNA's double helix is like a locked vault by comparison. And third, and most importantly, our cells are filled with enzymes called RNases. Their entire job is to seek and destroy RNA molecules once they've been used. After an organism dies, these enzymes are unleashed, and they can turn all the RNA in a body to molecular dust within minutes or hours. For years, the scientific consensus was that RNA simply couldn't survive the test of time. But then, we found the exceptions. Just as some environments can preserve DNA, it turns out they can do the same for RNA. Two places have emerged as havens for these fragile molecules. The first is the deep freeze of permafrost. The permanently frozen ground of the Arctic acts as a natural freezer, slowing down the destructive chemical reactions to a crawl. This is where Yucca, the famous 40,000-year-old mammoth, was found. The second is desiccation, or extreme dryness. By removing all the water, you stop the destructive enzymes in their tracks. This is how scientists were able to recover RNA from a 130-year-old Tasmanian tiger specimen stored in a museum. Even in these ideal conditions, the ancient RNA is incredibly fragmented and exists in tiny quantities. Getting it out is a high-tech heist. It begins in a specialized clean room, where scientists wear full protective gear to avoid contaminating the sample with even a single speck of modern dust or a skin cell. They use delicate chemical processes to coax the shattered RNA fragments out of the ancient tissue. Then comes the magic of next-generation sequencing. These machines can sequence millions of tiny fragments all at once, generating a massive amount of data. But this data is a chaotic mess. It's a mix of the ancient RNA you want, plus RNA from bacteria that colonize the body after death, and maybe some from the researchers themselves. So how do you find the real signal in all that noise? This is where the computational gauntlet begins. Scientists have developed a powerful toolkit to authenticate the sequences. First, they look for damage patterns. Ancient molecules get scars from the passage of time. One key type of damage is a specific chemical change called cytosine deamination, which causes a C base to be misread as a U. Seeing this distinct pattern is a strong sign that a molecule is genuinely ancient, not a modern contaminant. Next, they use powerful software to sort the sequences. They compare every fragment against huge databases of known genomes to filter out anything that's bacterial, fungal, or modern human, and keep only what matches the target species. But the most compelling evidence is biological. If you've extracted RNA from ancient muscle tissue, you should find high expression of genes related to muscle function, like actin and myosin. If you find that signature, you know you're looking at something biologically meaningful. This brings us back to yucca, the 40,000-year-old woolly mammoth. The study of its RNA stands as one of the greatest achievements in this field. After extracting RNA from its incredibly well-preserved muscle tissue and running it through this entire process, the results were astonishing. First, they confirmed the sample was authentic by finding a clear signature of active muscle genes. But they found something more specific: high expression of genes for slow-twitch muscle fibers. These are the fibers used for endurance, painting a picture of mammoths as long-distance roamers, trekking across the vast, cold plains of the Ice Age. Even more dramatically, the researchers detected the molecular echoes of yucca's final moments. They found active genes associated with metabolic stress. This molecular evidence aligns perfectly with paleontological findings that yucca was attacked by a predator, likely a cave lion, shortly before its death. We are literally reading the molecular signature of a life-or-death struggle that happened 40,000 years ago, and the discoveries didn't stop there. By analyzing RNA from the Y chromosome, they proved that yucca, long believed to be female, was actually a male. The mammoth isn't the only story. In 2023, scientists sequenced the transcriptome of the extinct Tasmanian tiger, or thylacine, from a 130-year-old museum specimen. The last one died in 1936. From its dried skin and muscle, they reconstructed gene expression profiles. But more importantly, they discovered hundreds of thylacine-specific microRNAs. These are tiny RNA molecules that act as master regulators, controlling which genes get used. This gives us a window into the thylacine's unique biology that its DNA alone never could. Pushing even further back, a 2019 study recovered RNA from a 300-year-old canid puppy from the Siberian permafrost. They found intact microRNAs that are specific to the canid lineage. One of them is known to regulate carbohydrate metabolism and starvation responses in modern dogs, giving us a functional snapshot of that ancient puppy's physiological state. So where does this incredible technology go next? One of the most exciting frontiers is paleovirology, the study of ancient viruses. Many of the world's most notorious viruses, including influenza, Ebola, and coronaviruses, have RNA genomes. Because RNA degrades so quickly, their deep evolutionary history has been a mystery. But now, by sequencing all the RNA in an ancient sample, a technique called metatranscriptomics, we can hunt for ancient viral sequences alongside the hosts. The Tasmanian tiger study, for example, found traces of RNA viruses. This opens the door to reconstructing the genomes of ancient plagues, tracking how they evolved, and perhaps helping us understand and predict future pandemic threats. And then there's the audacious dream of de-extinction. Companies are already working to bring back the woolly mammoth and the Tasmanian tiger by editing the genomes of their closest living relatives. But having the DNA sequence is like having a list of car parts. It doesn't tell you how to build the engine or tune it to run properly. Paleotranscriptomics provides that functional roadmap. By showing which genes were active in which tissues, it gives us the instruction manual for how a mammoth's body actually worked. This information is critical for any serious attempt to resurrect a functional, living creature. We are witnessing a paradigm shift in how we see the past. We're moving from a black and white sketch, drawn from bones and static DNA, to a vibrant, full-color moving picture painted with the dynamic hues of gene expression. The whispers of ancient RNA, once thought lost forever, are finally being heard. They tell us of Ice Age struggles, the unique biology of vanished predators, and the ancient viral enemies our ancestors faced. The journey into the paleotranscriptome has just begun, and it promises to completely reshape our connection to the deep and ancient story of life on Earth.